Zahira from Group A8 presenting our poster titled Recent Update on Pyruvate Kinase Isoenzyme, Distribution, Regulation and Clinical Implications. This presentation synthesizes key findings investigating how pyruvate kinase operates across different tissues. To start, pyruvate kinase is the crucial enzyme that catalyzes the final ATP gathering step of glycolysis, converting phosphoenolpyruvate and ADP into pyruvate and ATP. Every mammalian tissue contains specific isoform of this enzyme. Looking at cellular distribution, research show a clear division of labor in the liver. Parenchyma cells or hepatocytes exclusively contain the L-type pyruvate kinase. On the other hand, Kupffer cells are highly enriched with the M-type isoenzyme. Because Kupffer cells also exhibit over 30 times higher peroxide activity, it highlights their specialized antimicrobial phagocytic function. This distribution strongly suggests that gluconeogenesis is strictly confined to the parenchyma cells. Now, let's pass it over to member 2 to look at how the L-type enzyme is regulated.
Thank you, Zuhaira. I will be covering the regulation of the hepatic L-type isoenzyme and how it integrates our metabolic pathway. The L-type pyruvate kinase show classical allosteric behavior. It displays sigma kinetic toward its substrate. PEP is activated by fructose 1 biphosphate and is inhibited by both ATP and alanine. An incredibly unique finding is that this enzyme exists in two interconvertible redox form. When exposed to oxidized glutathione or oxidized mercaptoethanol, it then shifts into an oxidized form with a significantly reduced affinity for PEP. Adding a reducing agent completely restores its normal kinetic. This indicates that the thioredox state serves as a major physiological switch. This links perfectly with what we learned about protein and carbohydrate metabolism. L-alanine, which is a primary amino acid released during muscle proteolysis, allosterically inhibits L-type pyruvate kinase by shutting down this step. It safely diverts PEP away from glycolysis and toward gluconeogenesis. This dynamic forms the biochemical backbone of the glucose-alanine cycle, showing how amino acid catabolism directly controls glycolytic flux. Furthermore, this thyroid regulation relies on glutathione homeostasis, which itself depends on a steady supply of amino acids like cysteine, glutamate, and glycine. Next, member 2 will take us through the M-type pyruvate kinase.
Thank you, Bayani. While the M-type pyruvate kinase was historically classified as non-allosteric, recent evidence completely overturns that assumption. In leukocytes, the M-type enzyme actually has high-base allosteric inhibition by alanine, with cooperative PEP kinetics emerging at higher pH levels. This inhibition can be fully reversed by fructose 16 bisphosphate. Similarly, the liver M2 type kinase shows pH-dependent sigmoidal kinetics. Interestingly, at physiological free Mg2 concentration of 1 mm, the enzyme affinity for PEP drops substantially. It turns out that its inhibition by ATP is largely Mg2 dependent, meaning it might not actually be physiologically significant in vivo. We also see variation within the same organ. In the rat small intestine, two kinetically distinct populations of the M2 type kinase coexist, one that responds to fructose 16 bisphosphate and one that is completely unresponsive. Ultimately, this shifts our entire conceptual framework. Both L-type and M-type enzymes pose highly complex regulatory properties governed by substrates, metabolites, pH, divalent cations, and redox states, while the monad Weyman and Cangues model offer a great framework. Sequential, conformational transitions are needed to fully explain how these distinct activator binding sites interact. I will I will now hand the floor to Member 4 to discuss the clinical implications.
Thank you, Ain. In conclusion, pyruvate kinase deficiency in red blue cells severely impair their source of ATP, leading to hemolytic anemia. The observed kinetic abnormality such as include PEP, affinity, and a loss of cooperativity are driven by a shift in the enzyme equilibrium toward the active state caused by secondary negative SH oxidation, leading that the primary genetic difference alone. There was proof when macrotonic treatment successfully restored normal enzyme kinetic. Consequently, future research should investigate this to reduce regulation in non-hepatic tissue and explore its role in other red blue cell enzyme deficiency, highlighting pyruvate kinase role as a master metabolic integrator balance glycoside metabolism and reduce hemostasis. That's all for us. Thank you.